Standard School. Last time we have discussed about the basics of membrane transport, the passive transport. Today I will be discussing a few MCQs to reinforce these core concepts. So start with the first case. The first question is, which of the following is an example of passive transport? The answers given are the sodium glucose, SGLT, T1, the voltage gated channel, sodium potassium pump, and endocytosis. So I will repeat the question once more. Which of the following process is an example of passive transport? Option number A, SGLT1. Option B, voltage gated channel. Option 3, sodium potassium pump. Option D, endocytosis. The correct answer is voltage gated channel. We'll come to this. So the process of passive transport, the first and foremost is simple diffusion. Simple diffusion means soluble substances like oxygen, carbon dioxide, which are non-polar, that can pass through the lipid membrane. That is the simple diffusion. Whereas larger molecules like glucose and also charged particles require facilitation. It is passing through certain proteins which are called channels. These channels, as mentioned earlier, it can be ligand gated channel. It is a protein channel to which there is a receptor is there. On the receptor, a chemical which can be a hormone or which can be a neurotransmitter. Once it attaches, configuration changes occur and the channel gets activated and the ions, sodium, potassium, etc. can get inside. Again, there is no use of energy here, so it is an example of a passive transport. So the correct answer is, we have as mentioned, it is the voltage gated channel. So what is a voltage gated channel? These channels can be either a ligand gated channel or it can be a mechanically gated channel or a voltage gated channel. Voltage gated channel means when the membrane potential becomes a particular level, for example minus 60, this channel, there is a gate is there, that gate opens. So that gate is sensitive to voltage. So that is what is called a voltage gated channel. Some of the channels are always open and they are called the leaky channels. So hope you understood the concept of ion channels. So I'll explain one more, ligand gated channel. So this is the NMDR channel. So here you can see clearly there is a binding area for the receptor glutamate. When to this receptor glutamate gets attached, the channel gets activated and there is entry of calcium occurring. So this is example of a ligand gated channel. So what is a ligand gated channel? It is either a hormone or a neurotransmitter like glutamate, acetylcholine attaches to the channel gets open. Hope this is clear to you. Now we go to the question number 2. Osmosis is the passive movement of, option number A, ions across a membrane, water molecules across the membrane, large proteins across the membrane, gases across the membrane. Correct answer is water molecules across the membrane. So let us see what is osmosis. Osmosis is the passive movement of water across selectively permeable membrane. Depends upon the concentration of the solutes. So here you can see there is a semi-permeable membrane. Excess solutes are there on this side and the less solutes are here. So here the concentration of the solutes are more. So what will happen? Water will or the solvent will go to the area where there is excess of solutes. This is osmosis. Regarding osmotic pressure, we will discuss later. Question number 4. Leaky channels are ion channels that are tightly regulated by the cell, closed all times, open all times, only open during the active transport. So what is a leaky channel? Leaky channels are specialized proteins which will allow transfer of an ion. So this is a type of a facilitated diffusion. Leaky channels are always open. Typical example is the potassium channel. It always leaks. So the correct answer is it is always open. So the leaky channels are example of facilitated diffusion by the specific channel, and these channels are specific for ions. Some only allow potassium, some may allow sodium, like that. So here you can see the leaky channel. Potassium is leaking outside because inside the potassium concentration is high. This is in contrast to an active transport where energy is required by the ATP. ATP is split. So again, the leaky channels are example of facilitated transport maintained via special proteins or channels. Hope this is clear to you. Next question, glucose entry into the cell is by option number A, simple diffusion, ligand gated, voltage gated, active transport. Which is a correct answer? Of course, it's not a simple diffusion because simple diffusion occurs only for smaller materials like oxygen or carbon dioxide. That is simple diffusion. Glucose is a large molecule. It cannot pass through the membranes. So it needs facilitation. Facilitation is via channels. They are the glucose transportase, family of proteins called glucose transportase. So this is an example of this is a facilitation. So how it is facilitated? It is facilitated by the hormone insulin. The insulin attaches to the membrane and then only the channels become open or channels come to the surface. So entry of glucose into the living cells for their metabolism is via a ligand gated or insulin mediated facilitated transport. So this is a little more explanation. So here you can see this is the receptor for insulin. Insulin attaches to the insulin receptor that will induce insulin signal pathways, and that will lead on to activation of the glucose transportase, especially the GLUT4. This will come to the surface and attach to the membrane, and now the glucose can enter the cells. So what type of entry is this? This is an example of a ligand gated or facilitated diffusion. Is it active? It is not active because there is no energy required. Insulin comes and attaches to the membrane or the receptor that produces signaling pathways get activated and produces activation or opening up of the blood channels. Hope this is clear to you. A few points related to the diffusion. What are the factors affecting diffusion? The surface area is an important thing. The larger the surface area, the greater will be the, so surface area is important. Second is the gradient. The greater the, so it is always going depending upon the gradient. So higher concentration, more will be the, and the size of the gradient is very important. Larger the gradient, faster the diffusion process. Temperature is important. Higher the temperature, faster the diffusion, because the particles are moving because of the thermal energy. And the permeability of the membrane is very important. And finally, the solubility of the substance. The substances which are soluble in the lipid bilayer, that will pass easily, solubility. So these are some of the factors affecting diffusion: the surface area, the gradient of the material, temperature, permeability, and solubility of the substance. You should remember the Fick's law of diffusion. It states that in the steady state, the diffusion flux is proportional to the concentration gradient. So concentration is the number one factor that, so the higher the concentration, greater. And also it is proportional to the, directly proportional to the surface area, and inversely proportional to the thickness of the membrane. So we have discussed few MCQs by which we will be more confident in answering questions on the membrane transport, the passive transport. Please watch the video on passive transport also. Thank you.